Hello, everyone. My name is Huixun Du. I am from the lab of Daniel Weiner at the Buck Institute. I'm also a six-year PhD. Yeah, we do a really long PhD in the US, unfortunately. So today, I'll be talking about tension and gravity forces at play in aging. Oops, I guess it's not moving. Okay. So I'm sure at this point you guys have talked a lot about, heard a lot about cellular senescence and inflammation and immunosenescence. So basically, during the process of aging, senescent cells accumulate and immune dysfunctions occurs. And during this process, many age-related diseases show up. We as aging scientists often think about the chemical pathways, what's going AY during the process of aging. But there are also forces, physical forces that are at work all the time in our cells within the tissues. So these forces come from the liquid, come from the surrounding cells, also can come from the matrix that cells grow in. And as a matter of fact, during the process of aging, there are being documented literatures that some of the tissue environments can change. One example of that is tissue can increase its stiffness. We use this kilopascal. Oops, that's a pen. Oops, okay. All right. We use the unit of kilopascal to measure stiffness, and during the process of aging, the stiffness sometimes can double. And in age-related disease like fibrosis, this can actually increase many times. So in our lab, we're very interested in the question of how do forces modulate immune parameters like senescence and immune dysfunctions. And to do that, we actually have, so we actually study this in two different ways. One is through an increased force model, and second is we decrease the force on the cells and see what that do to the cell. So for the first part, we ask this question, can elevated tension in the form of increased stiffness facilitate cellular senescence? It doesn't work. Okay. So to do this, we have, this doesn't work. Yeah. We have cells that we can culture on different matrix, and we compare their phenotypes. So we use human lung fibroblasts, and we can culture the cells on the conventional way of culturing, which is on plastic, but it has really, really high stiffness that exerts onto the cell as well. Or we can have hydrogels that we measured it to mimic the lung, physiological lung tissue. We also have a stiffness at 50 kilopascal that mimics the fibrotic tissue level. And the next we do is we treat the cells with doxorubicin. This is an anti-cancer drug, but it's also commonly used in lab to induce senescence. And then we let the cells to grow in their corresponding stiffness environment. We let the senescent cells to develop. And at the end of it, we collect these secretomes for mass spectrometry. And we also do imagings, and we sequence the cell as well. So the very first thing we found is cells that are cultured on high stiffness plastic compared to the soft 2 kilopascal in condition, you have a lot of SA-beta-gal. So SA-beta-gal is a really commonly used senescence marker to identify senescent cells. These cells basically stain blue when they show this phenotype. And so here I'm showing the image that you can see with the 250 nanomolar doxorubicin, you have more senescent cells. We quantified this, and we also found this phenotype to reproduce in various doses of doxorubicin. So because we also perform mRNA sequencing, so we extracted the differential expressed genes of cells that are either cultured on this high tension condition or low tension condition. The first thing we did here is we did a correlation analysis. And we found that, not very surprising, that both the senescent cells that are cultured on either condition show high correlation to senescence. But what's interesting is on the right side, where when you look at these mechanotransduction related genes, only cells that culture on a high tension show stronger positive correlation. So mechanotransduction genes are a group of pathways that basically are responsible for transducing the force into the cells, modulating the cells through transcription or translation, and thereby reflecting different cell phenotypes. So what this is demonstrating to us that even in senescent cells, these mechanotransduction gene sites are at work. We further confirmed this and validated this in using a human data set. So we reanalyzed single-cell RNA sequencing from total of 66 human samples. Their donors either have healthy tissues, colored in blue, or have disease fibrosis tissue, colored in yellow. And what we found is mechanotransduction genes, they are all upregulated, as well as the senescent genes are upregulated only in these fibrosis tissues or cells. So what about the secretome? So because senescent cells make a lot of secretory phenotype, when we perform mass spectrometry, we found that these profiles are actually being modulated by tension. Here color in orange, green, and yellow is representing senescent cells. They're cultured on different types of stiffness. And you can see that they're basically clustering among themselves. And what we found is the reason for that change, or driving this profile difference, is the fact that high tension drives a lot of SASP. Here I'm showing a fibrotic condition compared to a physiological stiffness. Basically, cells are ramping up 80% of the SASP. And when you zoom in into those upregulated proteins, what you found is a group of mechanotransduction genes, mechanotransduction pathways, actin cell skeleton, integrin, row family. Okay. So that was the part when we have increased forces. Then what happens when the cells are actually in a state where the forces are absent? What does that do to the cell? To answer that, we actually thought about space. I know it's a little odd in an Asian conference, but it makes sense, trust me. So in space, our body are subject to less gravity, sometimes even zero gravity, right? And interestingly, astronauts who travel, these people that travel to space, even though they're really fit, but they come back with a lot of immune problems. And these features included increased viral reactivation, reduced T cell activation, and elevated pro-inflammatory cytokines. And that sounds awfully similar to an inflammation phenotype, right? So that's why we wanted to study how microgravity may be doing to immune cells. The way we do this on Earth is through a simulation of a rotating wall vessel. These cells go into this rotator, and this functions kind of like a bioreactor, but it actually has been used by NASA for many years. So basically, cells, when they're inside the rotating vessel, being rotated, and they are subject to different type of force vectors, and the force vector cancel out in a way that the microgravity effects is less. So you can think of it as cells are basically constantly in a free-falling state while they're being rotated. So we took fresh human blood, and we extracted the immune cells within the blood, PBMCs, and we subject the cells to either microgravity or to traditional 1G culture for 25 hours. And after that, we functioned the cells, we took functional assays, we also performed omics. So the first thing we did is we did single-cell RNA sequencing, and we found a bunch of genes and performed pathway enrichment on them. And what we found is a group of genes that are involved in the antimicrobial pathways, like paraptosis, interferon, are being downregulated. At the same time, genes that are involved in interleukin, pro-inflammatory interleukin signalings, are being upregulated. So seeing that in a transcript level, we obviously wanted to validate it further in a protein level. So we then did, on the left, ELISA measurements of the cells. And you can see that microgravity condition makes the cells secrete much less interferon gamma. Whereas on the right, these cells, when they're in microgravity, they produce more interferon, interleukin 8, which is also a pro-inflammatory cytokine. Next we did is we used a transcript-based inflammatory aging clock called IH. This clock basically measures the level of inflammation, and it also strongly correlates to multi-mobility and frailty. And what we see is microgravity has significantly upregulated or increased the score, suggesting that these cells are more inflamed. And we also measured the DNA methylation on these cells, and and the cells, when they're because they're from the same donor, and then we put them either in a 1G condition or in a microgravity condition. That's why the dots are connected. And using this DNA methylation clock, pace of aging clock, we found that microgravity on average elevated 4% the pace of aging. So in summary, I hope I've shown you guys that changes, or hopefully I have convinced you that changes in mechanical forces influence age-related immune parameters. And when there are too much force, in the case of a high tension environment, it induces SA beta gal, induces mechanical transduction, and also makes more SASP in these lung fibroblasts. And in the case when the force are actually lacking, microgravity, you actually have a suppressed antiviral response. You have elevated steroid innate immune inflammation in the immune cells, and both of which definitely demonstrate that mechanical transduction forces are very important for cell health, as well as it's relevant in the context of aging. So hopefully in the future, hopefully our continuous study of this subject will help us find some potential markers to rejuvenate force-related dysfunctions, such as fibrosis and many other actually related diseases, including atherosclerosis. So with that, I want to thank my mentor, Dan Weiner. I also want to thank my lovely Weiner Lab members. I want to thank all our collaborators at the Buck and our co-authors for helping me and finishing this work. I also want to thank the organizers for giving me the chance to present. With that, I'm ready to take any questions.
Okay, so we have time for a few questions.
Hi, great talk. Just two quick questions. So the first is, how do you disrupt that loop? Because the stiffness that the cells are experiencing from their environment are also influenced by the cell's behavior, right? Depositing, ECM, cross-linking things.
That's right.
So disrupting that feedback loop, I'm unclear what therapies might be involved in trying to disrupt that. And just the second question is that different cells in the body are obviously experiencing different forces. So those near bone are going to be experiencing different to those near lung. So have you looked at how aging compares in those two? And if they're different, why differing forces can have differing effects on cell aging?
Yeah. So first question on the matrix, right? So yes, cells do make their matrix, but it's also very cell type specific. Fibroblasts do not make like a lot of matrix. If you grow them on a different type of stiffness, it basically doesn't really modulate the environment they are growing in to that huge of extent to the point that you really need to worry about. So I hope that sort of answered your question. Second, yes, our tissues everywhere, organs have very different stiffness. That's definitely a caveat in the sense when we culture different type of cells, we need to actually adjust to what the cell's physiological environment is. And that's a huge problem in a lot of study. I think the field of bioengineering they pay attention to this more. They would actually grow cells in their corresponding more physiological environment because that can modulate the cell phenotype. But we sometimes, for the most time, actually sometimes ignore this question. Yeah, so it's definitely a good point.
Hi, thank you. Nice presentation.
Thank you.
I was wondering, are you suggesting basically that aging causes some form of microgravity in the body?
Yeah, so for the microgravity part of it, it's more of, actually, on Friday, there will be our collaborators talk, and he's also going to be talking about microgravity and how he basically cultured these organoids inside microgravity. But what we are proposing with microgravity is microgravity could be used as a simulator for some of these immune aging phenotypes, right? So we can use it as a faster way instead of aging the cells or aging cells in mice. We can do it using this way, yeah.
One more question.
Have you also analyzed other tissues, such as heart tissue or
Hack tissue?
Yeah.
Hack tissue?
No, heart.
Heart tissue.
Heart tissue.
Heart tissue, sorry, my apology. Yes, we did not. Actually, I did not, but Dr. David Furman, he did. So he has actually probably going to be slides on this cardiac organoids that he used. So I don't want to spoil alert, but basically it does something that's not good. Yeah, okay. Yeah. So last thing is, so our microgravity paper actually came out on Nature Communication this June. So please feel free to check it out. And the tension project is going to soon be on bioRxiv as well. So please connect with me if you wanted to know more. Yeah. Thank you.
Okay, thank you, Zoe. Thank you for the wonderful talk.